My name is Susan Ferguson. I'm an associate professor in the Department of Psychiatry and Behavioral Sciences here at UW. I run a basic science research lab, and my lab is actually based at the Seattle Children's Research Institute. So the Seattle Children's Hospital has a research institute downtown. That's where I run my lab. I'm also the director of the Alcohol and Drug Abuse Institute, which is where we are now. And so I'll tell you first, I guess, about my basic science research and then a little bit about what I do here. And so my lab focuses on trying to understand the cells and circuits that regulate drug addiction. And so we look really at all phases, from the initiation of drug use, the maintenance of drugs, that transition to addiction, and then of course looking at relapse, since that's one of the biggest problems that we see out in the real world, is that people that come off of drugs end up going back. And so we're using animal models, rodents, mostly rats, to be able to really look into the brain and try to understand the mechanisms that are regulating addiction, what in the brain is changing during that different course. Yeah, so I touched on this a bit. I mean, basically, collectively, the field of studying addiction has been around for a fairly long time now. Certainly, at least from the 50s and 60s, people were studying this, and we still don't have very effective cures for almost any drug, and even for things like opioids, which are probably our best treatments that we have, those treatments really don't work for everybody, and they don't seem to last for long periods of time. And so I think collectively, one of the biggest barriers that we still face with our research, and one of the things that we really need to address, is how do we move things forward and really develop lasting, effective cures. One of the big areas that I think we're going to see a huge explosion in behavioral neuroscience in general, and also with addiction, is doing a lot of single-cell sequencing of changes that are taking place. And so you really need big data to be able to analyze and process and understand that kind of those kinds of changes. And so I think it's going to really help drive our field forward in terms of understanding perhaps the underlying genetics of the disease. So we still, for drug addiction, we don't know genetically what might be causing addiction in certain individuals or not. And so even trying to understand susceptibility to addiction or resilience to addiction, we don't really know. And so what our hope is through things like RNA sequencing and looking at changes at the single cell level, we might begin to understand that.